So, shall we get started?
Yeah.
So today we will have like Veit's talk, and before we get started, I have just some quick announcements. First, as you probably know, this section will be recorded, so you can watch it later in our YouTube channel.
Não, é o Veit lá da Dinamarca que vai falar.
Beleza, Gilberto?
I'll also ask everybody to turn off the microphone. We can start questions. And also, I think it's better now to turn off the video cameras as well. And as I mentioned, questions will be in the end of the talk. And you can either, there is a button that it's raise your hand button, or you can also write your question in the chat. And it can be even in Portuguese that, yeah, we can read. And Veit can read too, because as you probably noticed, he speaks a bit Portuguese. Okay. Yeah. So, and the next week, we'll have a talk from Rafael Milani. He used to be Gilberto Dumont's student. So he's now in Neil Kellingham's lab in Chicago. So he'll talk about top-down proteomics, and the blood proteoform atlas will be quite cool. This is just like some talks that we have already confirmed. So Milani the next, and then we'll have also Giuseppe, Richard, which is also here, Danielle from Unicamp, and we'll probably see that most of it's mainly men that are talking, but somehow I don't know why, but the girls are more in the end. So we also have like Gianna, Juliana, they're from here, Brazil, but Kasia from US, and we are also inviting more people to talk. So let's talk just a bit about Veit. He, as you probably know, he's from Germany, Stuttgart, a nice place in the Bavaria region, right, Veit? So he's from Bavaria. It's not from Bavaria, it's just because I like to
You also looked to find the most beautiful photo from me, I can see.
It's really hard to get a nice photo from you, Veit. So later he traveled a lot. So he traveled a time to Cuba, where he met this nice girl here. I don't know what Ileana saw in Veit, but somehow they get married. And they even moved to Brazil, lived in Brazil for a while in Rio de Janeiro. So they speak Carioca. And you probably will see that Veit can't speak Portuguese, as I mentioned. And then later they moved to Denmark, where I met them. And they raised like this really nice family. Here's their young kid, Niels, which is not so small anymore. He's 10 years old, no, 7 years old, right, Veit? 9. Wow, time flies. Yeah. So Veit is like he's nowadays associate professor at the University of Southern Denmark. Published a lot of papers, so he has quite a good scientific track record. But he's also quite good bass player. So if you remember last week that I presented Simone, that he's like a drummer. Better than Veit, I must say, but Veit also plays really well bass. But I must say that he's not so good though in preparing barbecue. He likes to eat barbecue, but he doesn't know how to make it. But okay, without further ado, I want to invite Veit to give the talk for us. And please stay until the end, that we'll be open for questions. Thanks, Veit, for accepting the invitation. And it's really nice to have you here. Like Simone, you both are good friends of us.
Well, yeah, thanks a lot for the, I don't know whether it's nice or not nice introduction. I still have to think about that. You have to make a co-host, otherwise I can't show my slides.
No, you can. You can.
I can't share my, oh, now I can. Yeah, good.
Okay.
Yes, you can see it, right?
Yes, we can.
Good. Move this black windows. We're all already quite used to Zoom anyways. So thanks again for the invitation. It's a pleasure to give a talk in this interesting format. I also have to sneak into the other talks. I can see that it's not only people from the band, it's actually also other people. So sounds quite interesting. I have to say I actually missed Simone's talk, and it's quite sad because I had a meeting, and but it's very nice to see his logo at least, or his name on the list. So I will give today rather an overview of different things which are to be combined in the future and can be combined. So it's very high level at the start, but we go into some other details afterwards. So there are, let's say, 3 different elements I will talk about, which is mostly related to workflows to analyze proteomics data. One is automatic composition, which is a paper we published, I think, 2 years ago. It's quite interesting aspect and led us to look into how reproducible not proteomics data is, but the analysis of proteomics data is. And I will end with some of our apps. We have some apps where we can use to understand proteomics data a little bit better. Yes, so it starts with a high level. I'm talking about a typical data analysis of omics data, which is usually there are several similarities between proteomics, transcriptomics, and metabolomics at least. So we usually, I think you see my mouse. So usually we start with a study design, experimental design, where we design an experiment to study a biological system and try to find out more about it. We apply some omics technique on it, which is usually quite cumbersome experiment. Bioinformaticians usually take this experiment, put a small bubble or block on it, and that's it. And out of this experiment, you get some raw data, which then has to be pre-processed. There's some filtering, some QC going on. You have to, in proteomics, have to get the identity of the proteins, quantify them. And then you might want to do some statistical testing or some multivariate analysis because you have more than 2 different experimental conditions. You want to see what the patterns in the underlying data set are. And then you get some kind of subset of features, which could be proteins or transcripts, which show differences, for instance, between different conditions. You do some pathway network analysis, and then you get to some information, some inside the biological system, which then can be used to close the circle, because we then obviously get a few more questions we want to solve and dig a little bit deeper into the system. So more on the quantitative proteomics part and the viewpoint from my side. This is how I see a proteomics experiment or proteomics study. And I find it quite important already as a bioinformatician that the experimental design already decides about many things, and it actually influences everything downstream. So you should not take that too easy. You should think about having the correct experimental design to simplify your life afterwards. Otherwise, you might suffer. This is why I take that usually as a starting point. Then you get usually some biological samples somewhere from patients, for instance. You prepare these samples. Then you have, for instance, you do some suggestions. You have peptide mixtures, run them on the mass spectrometer. You get mass spectra of MS1, MS2, and also sometimes higher MS levels, which then you need to find the identity of the corresponding peptides, where we use, for instance, database search. Then the identified spectra, which then also are called peptide spectrum matches or PSMs. Then we need to extract some quantification, depends very much on the method you use. Want to do some statistics to find out what is differentially regulated, or some multivariate analysis to find patterns inside. And out of these, you get the subsets again, which then you can try to get some biological insight afterwards. I took this picture in a way that we often have, and that goes for the experiment and the data analysis. We have the data or the sample. We use some operation or some experimental process on it, and we get another type of data and sample. And I will come back to that later. So when we analyze proteomics data, there are different ways to do that, and I put them into 3 different categories. So in the end, I mostly talk about whole workflows. That means operations or software which can or which help you to get from raw spectra to biological insight. So there are quite a few available. So one category would be software suites, where we have the whole package in one. One quite prominent example is MaxQuant and PairSeries together, or Proteome Discoverer, the commercial version of, or one of the commercial ways of doing data analysis. They are usually quite easy to use, but are also, there is missing S, they are also limited in terms of what they can do, because in order to keep it easy, you have to put away the options. Another way to work with proteomics data or data analysis in general is to use workflow managers. There you can use, or you have available more different tools, and you can combine them often in a more flexible way. So you might want to try a different algorithm, for instance, and this different algorithm can be implemented or used in a very easy way. They are not as easy as to use as software suites, but they are still okay. There's the OpenMS NIME framework. There's Galaxy, which is becoming quite good also in proteomics nowadays. There are other workflow managers like Taverna, and there's also the transproteomic pipeline, which is also quite prominent, particularly in the States. If you want to really do your workflow your way, so you want to use a certain new tool, which is not in Galaxy, for instance, then you might have to tailor your workflows yourself. You have to find out which software you want to use, which functionality they have, whether they are compatible. And then usually we're making or using some script like Bash, Python, R, whatsoever, and put then these tools together to make a workflow which reads the raw files and gives us some output with, after specifying, for instance, the parameters. These are usually very difficult to reproduce. Why? Because in the papers they're usually described as in-house scripts, and nobody knows what they are. They're usually not published because they are written in a very dirty way. So this is a problem, because then the data analysis in a paper or a publication is not reproducible. And if you analyze this data your way, you will not get the same results. And also it can be very complicated to make a new workflow, mostly because software needs to be installed and can be quite tricky to be made compatible. I have to check the time. Yes. So when you want to do your own workflow, you want to assemble, you want to test a new tool in a workflow you might have built already, there are several questions we want to look into. For instance, one is, which software tool do you want to use, or which software tools you want to try? So there is a need of knowledge about what there is. We don't know all software tools. We don't read all the papers where a new tool comes out. And for that, we need a repository which actually provides this information about a certain number of software tools which can deal with different operations, different parts of proteomics data analysis. The other part is the compatibility. So one tool might spit out a CSV file, the other one MCML, another one is PEP XML. And then the next tool needs to be able to read these files. Otherwise, we have a problem. There are converters, but most of the time, quite often, they are a little bit limited or introduce some errors into it. So interoperability or compatibility between the different tools is quite important. And this information is preferably also available in such a repository. Then the next question would be, how hard is it to install and configure the software? You might all know or have had this problem that you want to try a new software and you failed already while you were trying to install it. This is a big problem. This is mostly due to dependencies because softwares depend on packages and libraries, and every computer is having a different environment, and nobody, or it's very difficult to program a software which can deal with all of these, and you don't get a new publication if you do so. In that sense, there are now some solutions coming out and they're becoming more and more popular. So one is software containers, which are built on Docker. So this is an environment which is like a small virtual machine, and then you have full control of how they behave because they behave all the time the same. And the other one would be software packages, which are done with Anaconda, and in bioinformatics with Bioconda way, which means this is a Python environment which allows you to install packages with exactly the same libraries in the background, and so they will work the same way on many, or let's say usually at least all Linux and Macs, and often also Windows computers. So then I'm coming to the last question, which is, let's say we now assembled already our workflow. It works, we get some data out. We don't know how good the data analysis we now did is. So we don't know whether the results are actually better than just using a different tool. In that, we need to benchmark and we check the results, and we also need to see how reproducible they are, because we want to show this result somebody else and want them to use your pipeline the same way. This is okay when you look for different tools. For instance, there are quite many benchmarking studies on database search, but it's not really available for entire workflows, and the comparison in workflow is a quite complicated matter. So I'm now going to the first part on the repository. When you're making these questions, you usually in a talk, you usually already know about the answers. So I'm also part, but it's quite a big community. We built in a European effort, we built a resource of bioinformatics software, or we call it software in the life sciences. This is called BioTools, and there are now over 17,000 tools described. So this is quite a nice number, which accumulated within the years. This is interesting for the users. We can see what there is. And it's also interesting for developers, because then if you want to have your tool more prominent, you might upload information on it, which should be very easy to use. It is still good, improvements are still possible, but it's quite okay to upload that information. And so each tool is displayed as a tool card with certain information. I won't go too much into details. And we hope that this catalog will stay there for a longer time. There's continuous development going on. We have funding over the next years and further on, as this is part of the Elixir infrastructure, which is a European infrastructure meant to be there forever. Might be a little exaggerated, but let's say at least for the next 10 years. When it comes to proteomics, then we also have quite good coverage. We worked on that the last 3-4 years. So we now have over 900 proteomics tools which are annotated and described in the BioTools repository or catalog. Why are there so many? So there are several reasons for that. We had like 200 plus tools from a colleague who already annotated them on this msutips.org website. We managed to engage the community because you don't want to annotate 900 tools and describe them. because you have to actually read these 900 papers. So it's very important to engage the community and have them annotating their tools themselves, and first have them understanding that it's actually a good thing to have their tool on there. And we seem to have managed to do that. That was also done by having some hackathons and workshops on, for instance, this conference, the Windows School 2017. What is in BioTools is very related, and particularly in proteomics, with this website. We have annotations. Annotations come from the community, different organizations, developers, and certain curators, which would be somebody like me and other people who are doing that more full-time than me. And these tools are then described by an ontology. I will come back to that. And what we hope that with this information about the tools, which is also described in a fair manner, we can help bioinformaticians so they know more about tools, what they can find and use, and also end users because they can discover new alternatives. So how is the tool described? There is quite a large set of descriptors, and we have a whole schema for that, which is built on JSON. One important part of the annotation or the description of different tools is the EDAM ontology. So an ontology is a way of describing things like anything like the Go ontology by terms, which get an ID and synonyms and so on. So, and with that, we can be more consistent in describing something because we have a controlled vocabulary which is better than keywords, because when you use keywords, then there are many similar ways of describing the same thing, and we do not want them. So when we have a tool, for instance, there's a topic, which in our case might be proteomics. There's an operation. So this is what the tool is doing, which might be database search. There's the data, and the data comes as what the data type is. And the data type might be, for instance, a sequence, for instance, a protein sequence in a FASTA file. And this protein sequence, and this protein sequence comes in a file, and it's the file format, which would be a FASTA file. So we have 4 different descriptors and identifier, which are then can be used to describe that software, and we can have also multiples of them. And when taking that, then you can say, okay, we have a tool in a simplified way, which has an input data, and this input data is, for instance, sequences, like in a FASTA file, which is the format. Then we have operation, so there's obviously also spectra somewhere. We have an operation like database search, which then makes out of this input, which would be mass spectra and the sequences, gives an output like identified peptides. And these come again in a file format, which would be, for instance, MCID or any other format. And this is quite interesting because if you have an input, an output, an operation, if you have a chain of tools working on your data, then this output becomes the next input for the next stage. And what we need is that this output needs to be the same as the following input for the next tool. Otherwise, we have incompatibility and we get into trouble. Or at least we have to work in converting the files, which usually is not a big not very fun. So what we did is then we took this information, not about the 900 tools, we made a proof of concept. So these were, I think, 25 different software tools, which it's a long time ago, we had to annotate and by this EDAM format. And when having these tools, we then wanted to see how many workflows or what would be the workflows you can automatically generate out of these tools, and assuming that they have to be compatible. So that they need to have a compatible output and input format if you have one tool coming after the other one. This was done with a software in collaboration with Annalena Lamprecht, which is called Prophet. And this Prophet suggests then different combination of software tools, which we then call workflows, and then had to implement them, which was not easy, but could be done. And then we run these workflows on some data and compare and see how different the actual results are, applying different workflows on the same data. So how does that work? So this Prophet, it's actually called now APE, as time goes forward. This software is taking all these tools, you do some constraints, and it suggests a certain number of possible workflows which convert your input data, which would be, for instance, WAR files, into output, which would be identified peptides or quantified peptides, depending on what you want to do. And here we have some PDFs too. And you can see the number of combinations you can have is that we have a combinatorial explosion, quite many, particularly when you have format converters, because everything can become everything. What we usually do is we want to involve the minimum number of tools, because less software tools means also less problems. So we took now the output of these Prophets and we implemented some of these workflows. So for instance, we took a case where we did, that was not quantification, it was identification of peptides. It was a data set which had quite many PTMs with a ground truth. That means that it was known what is in there. And what I find also interesting by using this ontology as a background, with that we can also write the method section quite easy, because we use these terms or these EDAM terms from this ontology to actually describe what the software is using, what's the input and output. So this also makes it much easier for somebody to read the method section, or even make the computer read it and automatically extract the workflow. I don't say it's very easy to do, but at least it becomes more reasonable to do so. What is missing here definitely, which we found afterwards, should be the versions of the softwares, of the tools, because that is quite important too. So when we now go to the results and look at different workflows and compare the results, we did this one, for instance, in the Taverna workflow manager. So I can describe it rather shortly. We took some raw data from some mass spec experiments. Then this is converted into MCML from thermo raw files. Then we identify the peptides using the Comet database search engine. Afterwards, we need to validate. That means we need to do some statistics to see which peptide spectrometries are actually the ones which we can trust. Afterwards, we have another validation on the protein level and see which proteins actually are identified. And then we have some filtering and some conversion we had to do to extract the UniProt accession names, or the names of the proteins or the identifiers of the proteins. And afterwards, so this is a very simple workflow, we have 3 different tools which are doing pathway enrichment, which are GeneTracer2, gProfile, and RichNet. And just to give you an idea how similar results are, so I might have to zoom a little bit in. So the gray terms were found in 2 of the workflows, and the green term is the correct one because the input data set or the input experimental data was from platelets. So we assume that platelet activation should actually appear as a very prominent term. It did not very much. So while RichNet, for instance, did fail on that, you can see there's not very much overlap. So I hope also this is a very big motivator for you next time you're doing pathway enrichment or go-to analysis. Don't trust it too much. If you use different tool, you might actually get the right different results. When you look at the quantitative side, so we now changed a little bit more in the workflows. So we took a search-going peptide shaker isobar workflow, which is done from, these are from one lab, but it's from different lab. And we took a transproteomic pipeline as a different workflow. And then we took some iTRAQ data and did the whole workflow from raw data to quantitative proteins in 2 different ways. This is a study which is coming from cancer patients, 2 different cancer types, and we had 4 versus 4 patients. So we have different patients and we have 2 types of cancer. Now, when running these 2 workflows, we find that there and which proteins were quantified, there's a certain overlap. But you can also see one workflow finds 802 proteins, which the other one doesn't. This is quite a high number, so much about using a different workflow. What was also interesting was when looking at the comparison on PCA, principal component analysis, which is a simplified picture of showing the similarity between the different data sets and different data samples and data analysis workflows, then you can see that if you have the same color and symbol, this is then the same biological sample or the same patient, but different 2 workflows. And there's quite some separation on them. And if you have all the triangles, this is one patient group. I don't know whether it's muscle invasive or not. And the circles are the other one. And different colors are the different patients. So the good thing here is that the similarity between different data analysis workflows is higher than for different patients. But it's still quite separate. So by using a different data analysis workflow on the exactly same samples or same patients, you get a certain difference which is almost as different as using biological replicates. And that means also this is a little bit concerning, because if we use have the same data but using some different data analysis, you might get actually quite different results. And if you compare the 2 workflows directly, you can see there's a certain pattern appearing here, so they have a different way of quantification, which is even visible on the ratios. So looking at these results and getting more and more concerned, this is what we bioinformaticians do, we make people concerned, then it is, or there is something happening in terms of reproducibility and applying different data analysis workflows. And until now, we always talk about technical variance, which means we run the same sample on the instrument twice, we get some different results, that's technical variance, or biological variance, which means we have different mice and they obviously also are giving different results. So I say that on top of that, there's actually also computational variance. So there's a variance which we get by using different data analysis workflows. And if we use different one, we get different results. And might actually be necessary at some point to take that into account statistically. So what are the reasons for this, whether different ways or what different results we get from the workflows? One is parameter settings. Different softwares have different parameters and they interpret them differently. They have different algorithms, so they work in a different way, then obviously also give different results. Then the environment where machine, the computer you are using can also give you different results. There might be a random generator, or you have different library backing up your software. And there might be also some omitted components where something happens quite often, like my favorite one is that people forget to adjust for multiple testing when doing the statistics. So given all these things, when we have different workflows, then we get these differences. Usually we do not, because everybody's happy when they get the results. And this is then a very biased result. It's like running your samples on one mouse and saying, okay, this is valid for all mice. So our way of looking into that a little bit and taking it quite simple still was trying to still keeping the, let's say, one replicate way, but at least making the reproducibility as as possible. So in the sense that if somebody in a different lab and a different computer is doing the same data analysis on the same data, they should get the same results. And this means you obviously have to apply the identical workflow. You need to provide a certain limitation on experimental design. And what we did, we want to be user-friendly and make it one workflow which is very easy to use. So this is still not solving this problem of computational variance, but at least we're trying to get a little bit more into the reproducibility or the getting the results work the same in each computer. So how did we do that? We went to a meeting and we had a hackathon there. The meeting was actually in an ex-monastery. So we were hacking during the day, and during the night we were praying for better data. Unfortunately, it was not working. Still, data is quite rubbish quite often. But so in that sense, we worked on having a template or protocol template for workflows in general, where we can upload softwares and use them to get the results from this workflow in a very reproducible manner. And reproducible is because all these softwares are loaded into a software container, which is a virtual machine, a lightweight one with a Docker, and as interface to actually execute that workflow without having to know too much about this. We use Jupyter Notebook, which can actually be used even to add some buttons and rulers and so on, make life a little bit easier. So with that template, we worked a little bit hard to get it done on a standard iTRAQ TNT labeled experiment. The software is called Iceberg, which was, there's no link, it was published a while ago, I think last year. And it's using all the things you get. You have to convert the files into MGF files, and then you get the whole workflow through until statistical testing and get the differential express proteins afterwards. There's quite a lot of QC in, so we're trying to keep things interesting also along the line so people can check whether the data is actually in good shape or not. We implemented different ways of different experimental designs, because what happens quite often that people might have a pool for comparison or not, very much recommended. And quite often people have different iTRAQ or TMT runs, and then they have to be combined. And it's all included in that software. I have to speed up a little bit. So the software, we call it a one-stop shop. This is now the Jupyter Notebook. You can't see much, but you can check it out yourself. So get some buttons in there. You have an interface to define your parameters, upload your files, and then you can directly use it and get the output. This is the software that's used, and it should be quite easy to use. And we even have installer for Docker and the Docker images, which should work quite straightforward even on Windows. So using this isoprod, we get okay-ish results, which means we compare them. Obviously, if you have a paper you want to compare it to original study, we get a little bit more output, and we also take into account in a better way the different MS runs. This paper did not, and they missed several peptides, and they also did not do the statistics on all data, but only per TMT, which means that this protein was found by them to be differentially regulated, and it's not. So in that sense, we hope we actually provided a better way to analyze data than in many studies. I would not dare it's the best way, but at least a little bit over average. So there's some recent work we are doing on also tackling a little bit this workflow benchmarking by comparing entire workflows in a more straightforward way. This is again an Elixir project or implementation study. These are the people involved. We now build certain workflows for label-free data. And what was the motivation? Motivation is that we have a certain data or also software heterogeneity, so that many different software tools, and also the data can be quite different. So it doesn't mean that one software or one workflow will be the best for all data. There might be actually There are workflows which are better for this type of data and the other one for another type of data. So there are many different tools and algorithms, and the problem is that many of these workflows are not really easy to use. They're not portable, which means you're using them on different computers without many problems. And they're also not scalable. So if you want to run 100 samples, you might want to use a better computer or even a supercomputer. Then you want to have that without many problems. So what we did is we implemented now 6 different workflows, or actually 5, because Proteome Discover cannot be used on this workflow. And in order to do that, we use this Nextflow language, which is a very nice language to use, for instance, Docker images, and run them on any Linux-based computer. So this can be scaled up quite drastically. They are still in development, but you can try them out if you go to this website. It's all open to anybody. Actually, I can't count. This is 6. So what I want to show here just shortly here is that these are the processes which are run on all different files, and see that many things are run in parallel because it was a quite good computer. And by running things in parallel, we can accelerate the data analysis quite nicely. And with this Nextflow environment, you can actually also put things on a supercomputer and have like 100 CPUs running on the thing. It's quite straightforward, and this Nextflow I can only very much recommend that thing. It's not that easy to use, but as soon as you had found out how to use it, you can ship your workflows anywhere and use them on quite nice, with nice scale. So now I'm coming to the next part, which is going a little bit off workflow topic. So there comes the time where you do not want to have a workflow spitting out all your results and all your biological interpretation. Particularly from when you have the quantitative features, you have protein quantifications. You might want to check different ways of statistical testing. You want to see your clustering results and cluster them with different numbers of clusters. You want to use different tools for GoTerm or pathway enrichment, and you want to see the results and check them. So then you don't want to have a workflow which is doing everything which as a black box. You want to have a more interactive way or software doing that. Or you might also want to look into protein complexes, which will be the last part of my talk. Having this, we developed quite a lot of tools lately, which are so-called shiny apps. They were working on a web server, but you can also run them on your own computer. And they are highly interactive, so you can upload your data and look into different aspects. They're all available here, or as Docker images. So the first one is we published that a few weeks, I think, ago. This is called PolySTest, and there's a specific feature on this statistical testing. So for instance, if you look into these 2 examples, you have 2 different experimental conditions. And when you then have green versus red, we might have not have actually some values because your mass spec doesn't measure it, because you miss them stochastically, or because they're too low abundant, or because whatever made this measurement not to happen. Maybe the spectrum was too bad to be identified by a database search engine. And then it can happen that you have all these missing values, like you have only 2 versus 5 values. And then how do you do the statistics? There are very few methods to take into account missing values. And most of them are based on being absent or not. And this doesn't work if you have them all on the same level. So here we have a situation, for instance, where I would say comparing these with these, there is actually no big difference. Also, we have many more missing values here. And I hope you agree with that. On the other side, if we have a treatment where most of the values are rather high, and we have a control, we have fewer values and they are lower, and then it's more likely that these missing values are actually missing because the proteins are more lower abundant in these cases. And this case should give you a better, let's say, p-value than this case. And this is what we implemented using this MISS test. We implemented certain statistics which takes that into account. You can read the paper if you want to get the details. And we found quite robust to take into account different reasons why they are missing. And that's something which is quite normal for proteomics data, because we have many reasons why a protein might or peptide might be missing. So what we did, we took then this MISS test and we took some other statistical tests and compared them. Particularly we took Limma, which is quite established now, and some other tests. We also did a comparison to the standard t-test, which I usually do not recommend, and I keep that opinion. And what we also did, we combined these four into a common score, which is called the PolySTest score. So on artificial data, we did quite many different scenarios where the missing values had different origins, like this is given by this mu parameter, different numbers of replicates. I think we had like several hundreds of test sets. We want to check how the statistics goes on, and giving on the different point situations or different data structures, we found that sometimes this MISS test is not giving you very much, and you would more go for the Limma, for instance, and some other tests, while in some cases MISS test is really giving you a lot of power. So seems to be that MISS test is helping a lot, but it's not the best test for all situations, which is why we combine these different tests to get a common PolySTest test. So this is the blue one, says that's a number of features you can detect, but you also want to detect the right features. So in order to detect it, we also looked into the actual true false discovery rate, which means knowing which features or which proteins are actually the ones are changing. We know that because we have synthetic data. Then we should be in the green area, because then this is the false discovery rate is the one which is actually reported, or lower as we want to have. And it seems to be working quite well, that the combination is usually giving you better results, which is also, sorry, there are many pictures, there might be a little bit too much information, but they say that most of them are in the green area when you combine different tests, which is quite nice, because that means you can trust your data a little bit more when you combine them. So when looking on real data, we took a data set where we have differentiation into muscle cells from satellite cells. And when comparing then or looking into one pathway, this is a muscle pathway, you can see we've quantified quite many of these. And practically all, I think we missed one of them, were found if we differentially regulate between the differentiated and the undifferentiated state. And the good thing is, when using this MISS test, you actually have this is the first state with quite many missing because they are too low abundant. And usual statistical tests would just discard these proteins. And by having this MISS test, we will now include them too. So we have a quite larger coverage of this pathway and the regulated proteins than one would have usually using standard statistics. So let me check the hour. Yeah, as usual, we're taking a little bit longer. So another type of, so this analysis can be done using this PolySTest web service. It's highly interactive. You upload the CSV file and you can check different thresholds. You can visualize in different ways and so on and so forth. It was recently published. Another way of analyzing proteomics data is when you have many different experimental conditions, and then we use data clustering to find out which are changes or which are proteins which are changed the same way. And if they are changed in the same way, they might be part of the same signaling pathway, or generally have a similar reaction to the changes in the system. For that, we need to do some standardization. That's usually what happened. It's actually a Brazilian data set. We compare different snake species and the glycosides. One of you listening might remember that very well. And you can see, for instance, when doing the clustering, that the most of similarity in the found glycosylation here is between Botrops insularis and Botrops jararaca, which are quite similar anyways. So there are many different ways of doing clustering. This is called hierarchical clustering, and I will focus on this Faz-Semient because I think it gives or yields quite nice information. Hierarchical clustering is getting difficult to interpret when you have high numbers of proteins, and usually you have thousands, so then this picture gets a little bit too complex to interpret. So what we looked into is that usually people apply statistics and then clustering. So they filter all their proteins for the ones which show some changes somewhere and they cluster. Or they cluster the whole data set and do some statistics afterwards and try to combine them. So, and there are many other ways people are doing it, and that's one of the problems. So how to combine statistical testing and clustering? There's no best way, and you can see there are many ways I can show here, and all of them will give you different results. So what we implemented is a so-called VS cluster, or variance sensitive clustering, where we take into account the actual variance of the features. Usually clustering is applied on the mean, so they don't take into account variance. We did, and with that we hope to get a better solution and not having to deal with this combining statistical testing and clustering. So how does that work? We have k-means clustering, which says, okay, each protein is part of one cluster, color here. Then there's Faz-Semient clustering, which means that each protein is to some percentage part of one cluster, and you could usually take the highest percentage. If it's like near 50% or below 50% distributed over the cluster, we actually can discard these proteins, which is also good. What we did then, we took one step further and took now what is given by the circle size. We also took into account the variance on this protein. Because a protein or measurement is not a very specific number, usually there's an error bar on the error margin attributed to it. This can be displayed in two ways. We can show this as circles, like defined by the standard deviation, or we can show it via density map. So this is now just a very short algorithm. So we did the very easy thing. We changed that general parameter into an individualized parameter and then translated that using some algorithm from the standard deviation. Again, we have this hard clustering, which distinguishes between different clusters, like definitely. Here we have the soft clustering or the Faz-Semient, where we have a degree. Each protein is part of a pattern, or for instance, increase with time. And this is now that's missing the color, but this is the same picture as before. So very shortly is that by taking this new algorithm, now we managed to get a much higher percentage or proportion of actual significantly changing. We did that check with statistical testing of the features in the different clusters. So we can trust the results a little bit better because we have a higher percentage of actually statistically significant features in there. So this worked quite well. So these are the higher bars on the left, on the lower right. And we also found, as a usual way of measuring how good the clustering is, we found it's called the F measure. We found it to be higher in usual cases. So that's the standard measurement to actually show that the clustering works well. We did that for many different synthetic data sets, again, to check whether it works. Applied it to some data with DNA methylation. This is VS cluster. We get a much nicer shape of distinguishing different cancer types. It's like, I think it was like 12,000 or 14,000 DNA methylations. And metabolomics, a very small set of 250 metabolites. We could distinguish many different temporal patterns, and they seem to make a lot of sense with some exclusions. In proteomics, we applied it also to several data sets, and this is that one, and maybe just keep on this one. And what we found is that now we have more clusters, or the proteins in one cluster are more attributed or more belonging to only one goal term or one biological or chemical property than different clusters together. Maybe that's a little bit complicated to explain. So in the sense that when you apply this clustering, you get more uniquely defined biological interpretations towards one clusters and not over all different clusters together. So we can interpret these clusters a little bit better. That's microarray data, and I think that should be proteomics data. Good. So again, this is a Shiny app, so it's rather easy to use and interactive, so you can upload a data set and do your stuff you want and get the results afterwards. So now I will move, let me check the time. I might take like 5 minutes longer. I look into protein complexes. So why do we want to look into protein complexes? So there are, I don't think I have to explain very much. There are protein groups which are stuck together by some chemical interactions, and there are quite many, and they can consist of 2 up to 82 different proteins. So there's a certain variety, and they have a lot of to do with signaling, regulation, and so on. So they're quite important factors in our cells. So this is now the ski complex. There's a head which seems to be detached. There's a body, and there are 2 skis. So we have a stoichiometry of 1 to 1 to 2. And this is its structure. Might be not true, but there's actually a real ski complex, and it has also 1 to 1 to 2 stoichiometry. It's quite interesting to see afterwards how these proteins behave as together and individually, and see whether we can extract some information about this complex from the qualitative behavior. So why do we do that? So there's now more and more evidence that protein complexes are co-regulated on the protein level, but not too much on the transcriptomics level. And this is quite important because most of the people doing biological science are actually doing transcriptomics and not proteomics, because it's too tough for them. And then the other paper says that we can get much more idea how protein complexes work from proteomics data than on transcriptomics data. And also the reason for that seems to be that in the cell, there's an active degradation of proteins which are part of a complex usually, but they might be a little overexpressed. And if they're alone there and not forming the complex, then they might be removed from the system. What does that mean for proteomics data analysis? This is our chance. This makes proteomics data very unique. And this also gives us a very nice insight into the biology. So there's an additional level. We now do not go only from peptides to proteins. We go also from proteins to complexes. So there are several questions. You want to see which protein groups are tightly co-regulated. That means they are changing the same way between different cell types and states. You want to see which proteins are maybe only sometimes part of the complex, sometimes not. And you also want to see in disease whether there might be some irregularities in terms of complex composition. And I think proteomics data can tell you that. So there is quite some information about the identity of the composition of complexes. There are 2 main databases for humans, at least, is Corey and complex portals. They are not that comparable because they have a different way of annotating the complexes. Then, in order to look into that and the similar behavior of complex, we did not use any APNS or any of this interact home data. We only looked into quantitative changes of proteins. So we took the ProteomicsDB database. They have 100. They had now they have more than 200. They had at the time they had 149 different cell types, human, and try to look and compare whether there's any significant correlation between protein groups. One pattern is that most of that data actually consists of missing data, so it was quite hard to do any statistics and base things, which we did this time on randomizations. So we had to invent our own randomization scheme, and also we are using a way of looking for correlation where I use this fast farms or factor analysis, which I will skip that slide very fast now. So we now have software called CoExpressor, where we took the proteomics data, we did different randomization methods, we took the quorum information about which are the proteins of a complex, and then evaluated whether there's co-expression. The randomizations forced us to create quite many scores or randomized protein groups for all, and to take into account the missing values. We had to do that at different situations, so that was a little bit harder. Could be done. So now we get p-values for each complex and each protein, each complex for its significant changes. The software you can actually add or you can integrate ProteomicsDB taking any protein group. It's still, I think, the maximum of 35 proteins, but which these proteins are doesn't matter. You can check whether there's any co-correlation going on. We also have a different app which is called Complex Browser. Now you can take your own data set and do very similar analysis. You can also do some QC on the quantitative level, and then compare whether there's any co-expression of the proteins which are known in known complexes. There are some other stuff happening there as well, so you can also use it for some statistics if you want. Yeah, I'm flying now over the slides. I'm sorry for that, but this is just the example. So you can see the proteins are changing between 6 conditions in a very simple, similar way. They are part of the ARP2S3 complex. And the other correlations, you can measure different things. So now, in the last, I think, 2 slides, I will show you some example how to use that. So we took a data set which was quite nice. There are more than 9,000 proteins measured in breast cancer. That's where they have 9 patients per cancer subtype. And then we looked into one complex and looked and compared this data to transcriptomics data. So we found that the proteins of this complex, the PA700 complex, they are actually behaving very similar. There's one protein which does not, which might be it's actually not part of the complex or just is always behaving different. You can see here again, different colors are different cell types. You can see we have quite many different cell types in there. And you can also see from the string database that are known, this complex is known to be very much a complex. More interestingly is when you look into the co-regulations, we looked in the correlation between the protein profiles. Here red means higher correlation, so this is the one which was not behaving like the other one. You can see in general the correlation is very high. There are some groups which are correlated very similarly, like these 4, and that's probably a transient state or something else. So this is the cancer data set. This is ProteomicsDB, like general human proteomics data. You can see actually the same 4 seem to be very similar. It was very nice to see. If you look on transcriptomics data, there seems to be nothing really correlating. And that is again showing that the proteomics data is telling us so much more about protein complexes than transcriptomics data. So I want to conclude. I hope that I shocked you decently with this computational variance. There needs to be something done about it. We are working on this benchmarking, trying to do a little bit in that sense in the comparison, but you should never trust the data analysis too much. If you use different type of data analysis or workflow, you definitely will get different results. Then we worked on making reproducible and standardized and user-friendly pipelines. User-friendly in some stages, still not very much there, but it's also not very easy to do. We have a new treatment of missing values now on statistics. So we hope now to get a much better or deeper insight into biology by taking into account a large number of proteins. This is happening through interactive apps or Shine apps, so you can upload some files and interactively browse and analyze your data. And as last part, this is also usable on protein complexes, and I actually hope with our apps we can help people to get much more biological information out there too. Shortly, I'm part of the protein research group. We are about 60 people. This is a very old picture. It's not old enough to have TRAQ on there, but and many of you guys listening. So it's quite a crowd. You can ask TRAQ. It's quite fun to be there. We have quite many different people, and yeah, we also have quite nice machines, now also computers, by the way. There are certain people in the group and my group also involved in all this research and collaborations, and certain funding agencies are funding the research. With that, I would thank you and sorry for taking a bit longer. I will check now the chat or listen to your questions.
Thank you, Veit, Dankeschön, for interesting talk. Really, really interesting and shocking as well, I must say. Just before I open for questions, I just want to remind you that the video will be uploaded in YouTube. So if you want to copy one of the softwares that Veit mentioned in the presentation, you can check them later. So we are open for questions. Everybody can also turn on the camera now if you want. And yeah, either use the chat or you can raise your hand and make your question directly to Veit. While people are getting not shy, I'll just make a question for you. I wrote a lot of things here, but yeah, regarding this pipeline that you mentioned, I wonder if you have already implemented for DIA data, that independent acquisition.
No, we didn't, and we don't intend to be honest, because we are not having much DIA data there. So I'm keeping myself a little bit out of that. These pipelines, they need to be quite specific on the data type, so it has to be either labeled or unlabeled. Otherwise, you turn it into a software suite and you will be forever programming, trying to make everybody happy.
But does it accept any data labeling? For example, dimethyl labeling? I think so, right? You can check for Silac, for instance. If you don't have
might be interesting to implement that. If you give me a few.
So you don't have dimethyl label there.
You can search it and use it, but the quantitative analysis won't be automatically there. So you have to take the intermediate results, which are always there, and then analyze them yourself.
Okay, but just a final question. These workflows, you need to choose each one of them, or like you can run all of them and then merge everything and try to extract as much as you can.
So we are planning to have that available. So you can run them all separately. They're all based on Docker and Nextflow. So if we have these softwares installed, then it shouldn't be a big trouble. Just running them and waiting until they're finished. You need sometimes a little better computer, but you anyways need that.
Yeah, that was another question that I had, like PC time. So you need a lot of I mean, a personal computer might not be enough.
With 16 gigabyte RAM, you can get there. So, but if you want to do your label-free analysis with TPP in 20 minutes, you might need a little bit faster computer.
Great. Okay. I think in the chat, Marcela has a question.
Hi. So I was waiting someone to decide to do questions since the last time I make a lot of questions, and I still have a lot of questions this time too. So Veit, thank you for the nice presentation. It was really nice and really scary too. And then I have also one comment. I will start with this comment because now it's more, I can see more paper like related to RNA-seq analysis, or that they do the Python or they are pipeline. They are usually now writing the paper that you can ask them, the pipeline, to you try to perform the same way of the paper to have this reproducibility that you told. Do you think that could be also like until we don't have this more easily workflow to try to follow, that could be a kind of a new future like for proteomics that they could also start to put this pipeline available for people that want to try?
Yes and no. So making the pipeline software available doesn't mean it will produce you the same results. If you don't put them into a software container or virtual machine, it will most likely, or it might not give you the same results. That's actually, I don't know if it's already came out, but there's a paper coming out from, I forgot his name. He's actually a magician and proteomics guy, but I forgot the name. So he tried to reproduce some proteomics study from Nature paper with the help from the original people. He did not manage to get the same results. Impossible. And so you need to containerize. So you have to have it, all the software, the same versions being on a software container available. So on the other question of telling the journals to making that available, we actually devised already some guidelines and asked all the different journals, not really interesting. Why? Because journals are also very interested in people sending their little bit worse studies in terms of data analysis, but still with wide interesting content. And if you make this, then life very complicated to them and they have to work very hard and they will just choose a different journal. So this is also the same with public data. So, you know, we should now, or most of the journal is a requisite to actually upload your raw data, at least to Bright or Massive or wherever. But now these repositories are competing against each other to have maybe not too many demands on what the data is. Because if they ask, oh, you need to give the cell type in an ontology, you need to have the sample preparation, so on, then somebody will choose another one. Yeah. That's the part that it's difficult to get the whole field moving there. But on the other side, data is becoming wide open. So now people are most of the journals are asking you to upload your data to a repository. So it's slowly coming.
Yeah. Thank you. And then I had another question about this PolySTest, yeah, for the missing. Do you think that we can apply this now for general proteomics data, TMT? I think it's mainly isobaric. I don't know if you are using also for label-free and so on. Or you think that we need to search like for a percentage of missing data, missing value that we have in the data set and then choose to apply it or not?
In principle, it can apply to everything because in the end, the missing values, it doesn't assume the missing values to be of random or other origin. They can be of both. But if you have TMT and iTRAQ and have like an experimental design where you have your experimental conditions distributed unevenly, you create a very big bias, and then you will artificially get more missing values here than there, because one MS1 will generate you for all proteins usually the same MS1 you get a missing value. So you have to be much more careful when you run TMT or iTRAQ, because you should avoid having these types of unbalanced designs, but you should anyway avoid that.
And then I have two last questions actually. One is like in that scenario that you show, like that you have the same study using two different pipelines, that you have like approximately 50% identification common between the pipelines and around 25% that's like exclusive for each one of the workflow that you use it. Have you investigated a little bit more deep what would be like this difference? This difference came from like where? Like
the identification, you say, right?
Yes.
No, identification we didn't check, but it's quite well known already. There is quite a big difference. And maybe we could have probably optimized the workflows and being more similar to each other, but we didn't want to. On the quantification, actually, that was the figure I showed. That was this strange shape of half U-like whatever. There's a different interpretation of or different ways of quantification in the tools, and that led to this thing. But no, we didn't.
Great, thanks. I think Wagner asked for a question, right, Wagner? Nice to have you here.
Oh, hello.
Yeah. Hello. Nice to see you guys.
Oh, you have a bigger beard than me. That's unfair.
So, thank you for the nice presentation, Veit. It was very interesting. I'm sorry I missed the beginning. I had a connection problem here. So I'm not sure if you mentioned something about normalization and scaling. And I've seen recently that this point gives a lot of difference in the results. The way that you normalize the data can affect deeply the quantification results. So what do you suggest regarding normalization? Most people use just log transformation and that's it. But I've seen that logistic transformation and ArcSym transformation may give different results, sometimes for a better normalized data set.
Yeah, so I think you answered already. Your question is, they said sometimes. So if you have a workflow which should be generally applicable, then I would go for, I think what we usually do is just keep the normal, because most of the softwares already have some normalization included, which they think is the best one. And I think on the statistics side, we might or might not at some point just take the median. Whether this is the right choice of normalization or not, that depends very much on the experimental design and the differences between the samples and so on. So I think that's a dangerous thing of actually having these workflows like fully as black box and available. Just somebody says, hello, I run the whole thing and I get my data out and that's it. We still should look in the intermediate data files and check whether things are okay and do some QC. Otherwise, and see whether everything makes sense. So otherwise, you get a result and it might spit out whatever many regulated proteins, but you miss the right normalization because all things are completely different and the normalization just fails on reporting it correctly. So yeah, so we kept it simple in that way, but again, that's, I think these Shiny apps could help a little bit, that you look a little bit into the details, try to understand your data interactively, and then you have more feeling of whether you're on the right way or not.
Thanks.
Sorry, Simone has a question.
Yeah. Hello, Veit. So thank you very much for your presentation. I have a question. Maybe you said that. I'm sorry. I was thinking about what we quantify in the mass spec, right? So what we mostly inject is peptide, right? So not proteins. So all of your statistical tools, I was wondering if they do something, if they take into account the fact that one protein can be quantified by multiple peptides, and the fact that some results, some protein estimations can be done with very variable peptide quantifications and other with very consistent ones. And I was wondering if you somehow weight that and you give like better scores to those that have more consistent peptide results.
Yes and no. So usually I keep a little bit out of summarization. So we do not, so my statistical tool, you can use them on peptide level or protein level, but they don't combine like MSStats, for instance, and MSQROP, the peptide level, which I think it's, to my opinion, is that there are not too many proteins which have sufficient peptides to do the statistics. Which in the future, and some studies might actually be there. But what we implemented, and these were the two slides I was jumping over, we implemented this factor analysis or forms analysis, which is by Lucas Schell. And we implemented it also as a Shiny app, which is still not out, so you can't see it yet, where you're weighting the different peptides and then get the protein quantification optimized on basing on the weights. So it weights the peptides whether they follow the same trend or not, and the ones who don't, then they get a lower weight. So this is in microarray data already quite established and was introduced in the proteomics data, I think 2 years ago, and we implemented your own version on that. And actually we have this app, and we also use it for the complexes in complex browser. So we also calculate a complex expression value, which is then also based on this weighting, because not all proteins are all the time behaving the same way. So you might want to have a lower weight for the protein which is behaving a different way or might not behave like the complex.
Sorry, can I ask one more question very quickly?
Yeah, sure.
One thing about I have a question about lemma, which of course, you know, I learned it from you. So my understanding that lemma is a statistical software that was created with the microarrays, right? So when you can have a lot of observation and you can get a p-value when you look at the dot compared to the other dot. Now we have rows in a table, but the concept is similar. So can you use lemma when you have more than two conditions? Because I guess the idea was to compare A versus B, right, at first. But do you use it also as an ANOVA, for example?
Yeah, you can use ANOVA designs on lemma. So that's quite often. It's doing linear modeling, which is ANOVA. So you can design in there.
Great. Thank you. Thank you, Simone. Paulo is also here, and he has a question. Welcome, Paulo.
So first of all, thank you for the enlightening talk today, for enlightening us in our morning here, at least in Brazil. I would like to, first of all, I share the same experience as you, that there's a great variability among all the. So what you're seeing over there, I'm also seeing over here. And I've noticed that, I've noticed that the pipeline that you're putting together enables you to aggregate various search engines, various filtering methods, so on and so forth. And we're always testing tools over here as well. So a suggestion that, or at least the way we have done it over here, is that when we're going to test a new pipeline or a new modification in the tool, there are certain tests that we do to make sure that it's working fine, because just referring to a number of proteins or peptide identifications, we've learned that that's not enough. We might have some tools that could be identifying more under a certain FDR, but that's just some overfitting. So we do two tests. One is we do a search against a sequence database that has only decoy proteins, and supposedly we shouldn't identify any proteins. So just to make sure that that pipeline is running okay. And if it's doing okay in this first test, what we do is we do a second test where we have two layers of decoys. One layer is the decoys required by the software to do all the FDR statistics, so on and so forth. And there's another layer of decoys where I know are decoys, but the software doesn't know. So for example, if I identify let's say 1,000 proteins, and I'm working with a 1% FDR. So I can assume that there are 10 proteins that I don't know which ones they are, but they're false positives in my result. And there should be another 10 proteins that I know are false positive, but the software doesn't know are false positive. And we use that as a second measure to evaluate a pipeline. So this would be like the first step in evaluating if a pipeline really deserves to be considered in your this thing that is a pipeline of pipelines that you're putting together. And there's also a second thing that I would like to share with you, because even in pipelines that we consider are working accordingly, we also see differences, just as you are experiencing as well. And even though these pipelines are trustworthy, we were trying to dig in why there are so many differentially abundant proteins that one pipeline will say that are indeed differentially statistically significant as compared to the other, and vice versa. And after carefully looking into the data, what we noticed is that if we're using a cutoff of, say, p-value 0.01 or 0.05, what you name it, most of the differences, most of what is unaccordingly, is due to small differences in p-values or marginal cases. So for example, in the other pipeline, I'm having a p-value of 0.07 or 0.06. So when you evaluate these differences in pipeline, and we've never worked our way to this level of refinement, but it would be also interesting for you to evaluate a measure of distance that would not be how many differences or how many differentially abundant proteins are there, but indeed what is the difference in terms of fold change and p-values, because these differences might be very marginal, and you might be working on that gray zone that you could go like, okay, it's a zero, it's just a small difference. So that's what I would like to share basically with you. And if you have something to complement over what I'm sharing, maybe just to give some food for your thoughts.
So what we are doing, so we already implemented quite a few different benchmarks. So one would be also looking at quantitative differences of the fold changes from the different workflows. So actually, quite many of these you named are already in there. And the other thing we already tried, what we're trying to do with this workflows and the comparison is we're using this UPS data set, which is yeast with UPS proteins at different concentrations spiked in. So we know what is in there. And then we can measure a recovery rate and everything quite nicely. So that's our step, which we probably will do in the next 2 months.
Fantastic.
But it's very much, and it's a good discussion, because when you have a proteomics data set, whether it's one with golden standards or not, which are good markers or QC markers, this is not that easy to determine. And one way to do that is would actually be comparing different workflows and see whether they still give you the same results, because then you have more evidence that this might be true, because there are different ways of getting to the final results. So that's why I like this term computational variance, because one could in principle do a statistical test or error bar on which is coming from applying different workflows or different variations on them. But they're still quite far away from that, I have to admit.
Great, thanks, Paulo. Does anyone have more questions? Simone again.
Hey, can I ask a very silly question, because this has been very intellectual, so I'm gonna bring you down to earth. You're probably the best person to answer this question here. Don't you have the feeling that the tools that the vast majority of the proteomic community is using are not necessarily the best, are the ones that are the easiest to use, right? So in this sense, I really appreciate that you're working really hard on these shiny apps, And I'm wondering if, you know, what are your thoughts about it? Like, because like we just heard in this conversation, like some of these tools, they become really complicated and most people have no idea what is going on behind the algorithm. And then they end up, you know, working with software that make safe figures, right? So I don't know, it's not really a question. I was wondering if you have a comment about it.
I think it depends very much on the resources you have. Let's say you're running a normal proteomic study and you just want to get the results, then you probably have to go for an easy way and some tools which are easy applicable, which in most cases actually is done by using MaxQuant, and that's why in Bright most of the datasets are used for MaxQuant. If you have like this cancer data set I showed at the end of the 9 patients per 5 different cancer, like a huge number of samples, and they did fractionation to death, so otherwise you don't get 9,000 proteins quantified over all samples, and they had somebody who digged into that and worked on that, so otherwise they would not even publish their own next flow pipeline and stuff like that. But if you want to get that way, you need somebody. You can't, unfortunately, things until they are really usable and user-friendly, it takes some time. Mostly because we don't really get good publications from telling, oh, now this is user-friendly. So yeah. Sorry about it.
Right.
But then you have a citation of that, you know?
Yeah, it's true. No, and also the journals are going a little more. So if you have a really user-friendly way to do something, it's getting better. But it's also, as a bioinformatician, I'm more interested in finding a better algorithm than working on a software making it even more user-friendly. That's just boring. So it's very difficult to also motivate people to actually make a user-friendly software. That's thing for software engineers, but it's not research.
Yeah, but you think like that, but you know, people have you know, limited capabilities. Like I'm speaking about myself, of course.
Me too. You need this way. You need to combine who want to do your part and who would have the intellectual like part to make this like more user friendly. So
yeah, you're right, but it's like you're also researchers. You don't want to develop sample preparation and all the different steps and explain everything to that detail that everybody can do that perfectly and make diagrams here and there and so on. You don't want to do that either. You want to test a new sample preparation method and check whether you get 100 proteins more, right? So I just think the same like you.
But if you can merge, you can merge both. But if you can merge both things, better, right?
Yeah,
Always
yeah. And,
pursuing for the best algorithm plus making it easy to use. And myself as well. Like, if you do too complicated, it's hard to implement. At least
you will have the work because then we will send it to you on the data.
Yeah, but I still have to like read that email. No, but I think that Leonard Martins also said that several times that when you had some nice pictures I don't have, but so if you develop a new algorithm, it takes like 10% of your time, 90% of the time takes make it installable, that people can install in different ways and so on and so forth. That said, it's getting better because with Docker and Bioconda and all these things, it's getting much easier to install things. So we get rid of the installation problem. Where the software is easy to use, that's not, but I think most of the people actually fail in installing a new software they want to try out. And if that is the way, then we are already one big step to the next problem we have to solve. Right? Yeah. I assume that all of you already tried to install a software and gave up. So if that problem is solved, then we are getting better.
I remember Dante-R was a pain in the ass to install. I remember that we used to have a lot of problems.
Which one?
Right, Wagner? Wagner's like saying, yeah. Dante-R. That's the one that
yeah.
R for dummies.
That's not my fault.
Well, but at least you are making like a sexier figures, I must say. So you are pushing yourself harder to make like a more user-friendly interfaces and also sexier figures, which is cool.
Sexy is good.
Okay, any more questions? No? Okay, so I want to thank you again, Veit. It was really nice. And
Thank you.
You can wait that we are definitely going to contact you to try these things that you've shown for us. And of course, we will have questions, and you need to please help us. And
Go ahead. I am usually quite fast in answering emails, so
great.
there's trouble.
Yeah. And everybody that wants to also contact Veit, you can do that, and you can help. Oh, Diogo, is it a real place that you have behind you? No.
No. It's just a fake beat.
Okay, it's a fake one. How come the guy's like working in Vienna?
Oh,
you don't have this kind of beaches there.
No.
It's just a dream.
Okay. Yeah.
I have a vocab actually in the background, but it didn't work very well. No.
Okay.
Anyways.
Yeah, but thank you everybody, and please share the link of the YouTube for people that missed the talk and want to see it again and try all the cool softwares that Veit shown. And yeah, and again, I want to invite you all to join us again next weekend, Wednesday. It will be on the afternoon though, to watch Rafael Milani's talk. Thank you everybody.
Thank you. Bye.
Bye-bye. See ya.
Thank you. Bye-bye.
Valeu, Veit.
Hello.
Foi ótimo. Só a gente liga aí.
Você não muda nada, né?
Não muda nada, não fica mais velho, não.
Bom, aquela figura que o Thiago colocou, ela tá bem mais nova, né? Mas tudo bem.
Das war denn hier? Denke mal,
Vai lá. Manda um beijão para a Eliane e para o Nils, tá?